So today, oh, there we are. Jeez. Okay. Today what we're going to do is we're going to cover primarily B cells and nothing else. So this is the last of the major cell types that we're going to talk about with respect to immunology. So and the homework assignment kind of reflected that. We've talked about TC cells, we've talked about NK, we've talked about T helper, macrophages, and neutrophils as examples of phagocytes, and now we finish off with B cells. So the main focus today is just going to be to talk about B cells and their function, and if there is time, we'll try to do a couple of problems afterwards as well. The first thing I wanted to do, I suppose, was actually start with a quick little just review for all of us based on what we talked about Wednesday. And that the question is, what's the difference between a professional antigen presenting cell and a cell that we would just characterize as a phagocyte? All right, so again, what is the difference between a PAPC, professional antigen presenting cell, and a cell that we would just characterize as a phagocyte? So take a minute or two. I'd like you to talk this one over with somebody else. There's at least one difference, something that one of them can do that the other one cannot. And that's what I'm mainly looking for, but we'll try to generate whatever we can from this. All right, take a couple of minutes. I don't know if there's a knockout or Pseudomonas knockout library. Third way you could do it is take those tail fibers and blast them against other tail fibers and see if the receptor is known for those other phage which the tail fibers belong to. But you would need to have, and I'm talking like 99% identity, because I think most of the basic structure is similar.
Is there anything in the genome?
I know.
Like you told me to sequence the receptor and spike?
Well, spike is the tail fiber.
But most of it is for the actual structure, right?
Probably what's unique is just one small region of it. I would assume. That would be my assumption. And phagocytes. What makes a cell a phagocyte? What are some characteristics? I can see it's going to be that kind of Friday. Okay, performs phagocytosis. That's a great start. Do professional antigen presenting cells do this? Yeah, they do as well. And so this would be, again, if we walk through the steps, there's an attachment between a PAMP and a pattern recognition receptor on the surface of a phagocyte. We get phagocytosis, or that ingestion. The phagosome undergoes acidification, fuses with the lysosome, breaks down the pathogen. There's something different in how these two, though, respond afterwards, or what happens to that broken down product. What is the difference? Or what do one of these cells do that the other one does not? When we say antigen presentation, what do we mean by that? Okay, so say that again, Ellie. Okay, so you're saying, now do you mean for the recognition or afterwards? Okay. So professional antigen presenting cells, and an example of one is a macrophage. Macrophages, we'll prime this a little bit more. Macrophages present the pathogen on the surface, and they interact with what kind of T cells? Macrophages interact with which of the two sets of T cells that we talked about? Helpers. And helpers have what co-receptor? CD4. And CD4 interacts with what? What does it CD4 bind to? MHC2. Perfect. So professional antigen presenting cells are cells that have MHC2 or that express MHC2. Okay. Macrophages are one class of cells that fit under this. The B cells are another. There's a couple of others in here, like dendritic cells, which are basically specialized macrophages, will present as well. But these are the only ones that we consider to be professional antigen presenting cells. Okay? Professional antigen presenting cells, by definition, because they perform phagocytosis, are defined also as phagocytes. They are just a specific subclass of phagocytes. Okay. So what we're going to spend a little bit of time then talking about is how do B cells actually present pathogen on their surface and then allow for their stimulation by T helper cells. Okay. There's some similarity to what we observe with macrophages. So let's say that we have a B cell. B cells don't recognize, B cells primarily recognize pathogens with a receptor on their surface known as the B cell receptor. So whereas macrophages and the other professional antigen presenting cells recognize patterns, B cell and the B cell receptor, they recognize specific antigens or specific pieces of a pathogen. Functions very much like the T cell receptor. If that part of a pathogen is present, say it's a spike protein on a virus, this triggers then phagocytosis to occur, for that pathogen to be brought into the cell, broken down, and ultimately presented on MHC2. Presented on MHC2 then allows for what cells to interact with this? So who's going to come to interact with the B cell at that point? Good, the TH cells. And remember that we said that two things have to happen for the TH cells to be able to secrete cytokines. So one event that has to happen is the CD4 has to interact with MHC2. What is the other event that has to happen at the exact same time? What else must the T helper cell recognize? And if you don't know, take a look at your notes. We covered all of this on Wednesday. Yep. What else does the T cell have to recognize? That T helper cell, what does it have to recognize to be able to secrete cytokines? Not necessarily the infected cell. That would be a job of a TC cell. Focus in on what's happening here. What's happening here? Brent Taylor.
Is this the lock and key model that you were talking about
Yeah.
last time?
So what's the other?
PCR?
Yeah, it's the T cell receptor. So what does the T cell receptor recognize, Taylor?
It recognizes the piece of pathogen that's in that receptor.
Good. So the T cell receptor on the surface of that T helper recognizes the antigen that's presented in the MHC2. If these two things happen at the same time, the T helper cell secretes cytokines to activate that B cell, and the T helper, again, will secrete cytokines to activate itself. What does an activated B cell produce? Produces antibodies. Okay. When a B cell becomes activated, goes from a resting state to begin produce, one, it will begin to proliferate. So we get a bunch of B cells produced. Some of those B cells become what we call plasma cells. And the main function of those plasma cells is to just kick out boatloads of antibody. We're talking about 80% of the proteins that are produced in those cells is antibody. Now an antibody is going to be secreted from these cells. So they're actually going to be released from those B cells. And just to point out real quick, these antibodies, the receptors that they have on them that bind to the pathogen, this is actually the same structure, mostly the same function as the B cell receptor. The B cell receptor is, in essence, a membrane-bound antibody on the surface of that B cell. Okay? So it means that that B cell, the antibodies that it's producing, are identifying the exact same pathogens that the B cell recognized originally to interact with the T cell. Here's our basic antibody structure. I'll draw those in a different color. So the brown represents disulfide bonds holding together different parts of the antibody. The antibody has a basic Y-shaped structure. What I'd like you to do is label what this region is, what this region is. There's two of them. And then also identify for me where are the constant regions, so those areas that are conserved regardless of which antibody it is, and which regions are variable, meaning they differ between each different antibody, or they differ between each antibody produced by the different B cells in our body. So 4 parts: label, label, identify, identify. All right, take 2 minutes. Okay. What are these two arms here at the top? What are those known as? Or what's the term that we use to describe them? So the heavy chain, Allison, would actually be, I'm not actually asking you to identify those, but I will cover those real quick. So the heavy chain is, there are 2 heavy chains. Here's one, and here's the other. So the heavy chain makes up the majority of the body, and then we also have these two smaller light chains as well. Okay? That's just some basic, and this would be more important if we were actually talking about how does the antibody get made. We're not going to focus on that part, but sometimes those terms will be referred to when discussing parts of the antibody. What I'm looking more for is, what is this region or these two arms? What is their function? How might we describe them? Antigen binding site. So this is the region that actually binds to, say, a spike of a virus, for example, or to a outer membrane protein on the surface of a pathogenic bacteria. How many of them are there? 2. And I see why you would say 4. The light and the heavy chain can differ in structure, so it's more what the two of them make up together makes up one of these antigen binding sites. And this the reason why I bring this up is just to point out that any antibody can bind to 2 antigens at any time. Those could be on different viruses, or they could be on the same virus. Okay. What is this region that we have labeled number 2 called? Stem. Sometimes you'll also see the term FC for fragment crystalline, or I just would refer to it as fragment constant. And that actually gives away then which region of this genome is constant. So the stem, regardless of which antibody and what is the target of that antibody, this stem region is always, always, always the same. And that's really important because the antibodies actually help to link together the adaptive and the innate immune response. The variable regions are going to be those that give diversity to antibodies, and those that help to recognize the hundreds of millions of different potential antigens that could be recognized. And so these antigen binding sites, particularly right at the tips of these, is where we see the greatest variability in the genome sequence for these different antibodies. So antibodies can perform a number of different functions. I don't go into all of them in great depth, but our book refers to, for example, one of the functions is we say that they neutralize. Another term that we might use is they opsonize. And there are a couple of other functions that we won't talk about just because of the depth and time that it would take to go into them. But when we talk about neutralizing antibodies, we use this term typically when we're talking about viruses or when we're talking about toxins. So antibodies that have been produced to recognize toxins, if they bind to the toxin, we say that they neutralize or prevent the toxin from actually causing a symptom or an illness. If you've ever had the DTaP vaccine, which is diphtheria, tetanus, and pertussis, those 3, the vaccine itself is a toxoid vaccine, meaning it's made up of heat-treated or heat-fixed toxins from each of those different bacteria. The goal is to get antibodies produced that would help to neutralize that toxin down the road if ever you were to be exposed to it again. The other thing we say is that antibodies opsonize, and that's just a fancy word for saying antibodies coat. So when we say that an antibody opsonizes or antibodies opsonize a pathogen, let's say that this is a virus with multiple spikes. And we have an antibody that recognizes this spike. This antibody these antibodies have opsonized or coated this virus. Now, you may look at this and say, isn't that neutralization? And the answer is yes. these terms don't have to be mutually exclusive. Antibodies can be doing more than one thing at a time. It neutralizes the virus from being able to attach to a host cell because antibodies to these spikes would prevent the virus from attaching to the receptor on the host cells. But the second part is this opsonization is where we see a tie together between the adaptive and the innate immune response again. One of the things that we've already said is that each antibody has this constant region, or this stem. There are receptors on the surface of macrophages and neutrophils and even natural killer cells that recognize this stem region on an antibody. Okay. So these regions again are known as FC regions, and the receptors are known as FCRs, or FC receptors. So let's take a look at this real quick. So here is an example on the left-hand side where we actually see this, these FC receptors working in action. So we've got, in this case, staph epidermidis expressing, two different antigens or epitopes in this case. We've got antibodies that will bind to it, opsonize the pathogen. This FC region can bind to the FC receptors, and if this is a phagocyte, this allows for the phagocyte to grab a hold of the pathogen, not directly, but indirectly, and bring it into the cell and destroy it. Okay? So there's a lot of playback between innate and adaptive immune responses. We talk about them oftentimes as being two separate and completely distinct features, but actually they're working a lot together here. We see this also with, like I said, we see this with neutrophils, we see it with NK cells. The mechanism with NK cells is quite different because, as we talked about before, NK cells don't phagocytize. They will secrete granzymes and perforin to kill organisms. So we won't cover that particular mechanism with NK cells. Some bacteria, some viruses have ways to get around this mechanism. So we've talked about how some viruses will repress, for example, the expression of MHC1 and MHC2. We might also find repression of the expression of these FC receptors on the cell surface. Here's another example, though. Staph aureus produces a protein known as protein A. Protein A functions like an FC receptor. And so what Staph aureus can do is, if this protein A is being expressed, it binds to the FC region of the antibody, like is shown here. And what does that do? Or what does that prevent? So binding to the FC region prevents this antibody from binding to what? To the host cell. So in fact, what Staph aureus can do is it can sop up these antibodies, basically flip them around so that it might allow for aggregation of staph epi if this is attaching to more. But because it binds to the FC region, it basically is a competitive inhibitor of the FC receptor. And in doing so, then, it reduces the ability of the innate immune system to phagocytize Staph aureus in return. Okay. I'm going to pause real quick and just see if there's any questions with those features. Taylor.
So when the antibody in that second that we talked about, would that also prevent it from attaching to any other host cells, because the antibody would prevent it from attaching to other, like, epithelial cells?
Do you mean prevent the Staph aureus from attaching to other cells? I suppose it's possible. I think it would be possible to prevent it from doing so. You might see a scenario. The only way I could think around that would be, I mean, this is a antibody that recognizes a triangle region. It looks like. So if you had, let's say this is our antibody right here. If you had another Staph aureus that had the particular antigen that was recognized by that antibody, this one might be able to attach then. I would think that this kind of process here actually might be able to contribute to biofilm formation by helping to bring those Staph aureus cells together, clumping almost. But that's just a hypothesis. I don't know if that's really what happens or not. It could prevent them from attaching, absolutely. Okay, any other questions? Okay, so what's the key takeaway on B cells? What is their structure? What is their function? What features do B cells have that define, or that we could be using to identify one? B cell receptor, absolutely, that's one. And what's the function? Secrete antibodies. That's the main function. Okay. Yes, they can function as phagocytes, but that is, they are not doing that as quickly or as often as we find with macrophages. It's really a means to an end there. Where we're going with this in the next week, then, is taking a look at vaccination as a way to artificially induce immunological memory. So we've talked about that there are two parts of the immune response. There's the innate and there's the adaptive. The innate response to, if your body sees a pathogen a second, a third, a fourth time, the innate response responds to it the same exact way every single time. If you've seen the movie 50 First Dates, consider the innate immune cells as being like Drew Barrymore's character. Doesn't remember what the last date was like. Okay? The adaptive immune system, on the other hand, is Adam Sandler in that movie. Remembers all the history. Okay? The adaptive immune system, what we find with it is that over time, at the end of an infection, some of the cells that are produced actually become what we call memory cells. They are long-lived. We don't necessarily know how long. This is kind of up for debate. We don't know where they go, if they go back to the bone marrow or if they're just kind of free-floating through the body. But what we do know is that these types of cells are produced. And what we find is then on a second or a third or a fourth exposure to the same exact pathogen, the one with the same similar structures, our adaptive immune response responds to it much more quickly and with much more force a second or a third time through. This is the principle for vaccination. The whole idea behind vaccination is to expose somebody to an inactive or to a weakened form of a pathogen so that if your immune system sees it down the road, say a year, 2 years out, it's primed. It's ready to respond much more quickly so that the illness you experience is mild to very minimal altogether. So this image here is just showing, again, kind of that contrast, or showing, it's showing two different immunoglobulins or antibodies, but really what we see is, I want you to just focus on the second one here, IgG. This is the major antibody that our B cells produce when they become plasma cells. So here's our first exposure, and then whenever we're exposed a second time to the same antigen, we find those memory B cells beginning to proliferate rapidly, very quickly, mass-producing amounts of antibody to neutralize the infection much more quickly. All right, questions? Molly?
So, we're probably gonna talk about this more when we talk about immunization, but so when they're saying the first exposure in the case of, vaccination, that's like the vaccine, and then you're referring to the second exposure as potential exposure to the disease?
Yes.
And then where do like boosters come into that?
That's a great question. So boosters would be in cases like, I mean, we have to get, tetanus we think only lasts about 10 years or so, right? This is where the question of like, where do memory B cells go, how long do they last, this is kind of in debate. The boosters, some antigens are not very strong antigens, and what I mean by that is they don't trigger an incredibly strong immune response. And so boosters, the goal of a booster is to expose a second or a third time artificially as a way to enhance that memory. So
does
it would
kind of,
be
like, have a summation effect, like
exactly. So what you might find is like, here's the first exposure, booster might get a little bit more, third booster you might get a little bit more, so that if you ever are exposed to it, now you're up at this level by the time that you are naturally being exposed to it. So a great example here would be HPV. It used to be a 3-course shot, the initial and 2 boosters. Now it's just down to 2 because they've actually found that you only need 1 booster for long-term protection. Yeah, I guess that's the basic summary of it. So some things just aren't very strong immunogens. We have to ramp up the immune system, basically try it out 3 or 4 times before we get the effect that we want to, or levels of memory cells produced so that if we are exposed to that pathogen down the road, the disease is incredibly minimal. That's a great question. Any others? All right, we are not a lively crowd today. I think we can all be honest on that. Have a good weekend.